Hey Team Bio, today's screencast is about protein structure and the relationship between the structure of the protein and the function of the protein. So as you know from last night's screencast, the 3D shape of, the 3D shape of an enzyme determines whether or not the enzyme functions. And today's screencast is going to be about the 4 different levels of shape that every enzyme takes on. Or I guess every enzyme takes on 3 and some take on 4. Okay, so the first layer of structure is called the primary structure, and it is the sequence of amino acids in the protein. So the primary structure is the sequence of amino acids. And this is just a quick reminder for you all, but here, as you know, is the structure of a generic amino acid. We've got the amino group on this side, the carboxyl group on this side, and an R group in the center here. And the same is true of this amino acid. So here is one amino acid, and here is another amino acid. And when they join together, it's through a dehydration synthesis reaction where the OH group on the carboxyl binds with a hydrogen on the amino group of the next. And obviously, this makes 2 H's and an O, which is water. So this is a dehydration synthesis reaction when these 2 amino acids come together. Dehydration, synthesis. And this bond between the carbon of one amino acid and the nitrogen of another, it's called a peptide bond. These 2 amino acids together, when we have 2 amino acids joined together, we call it a dipeptide. Di meaning 2, and each one of these is also an amino acid or a peptide. So, and this reaction creates the dipeptide and also water right here. So if we were to do this reaction over and over again, this dehydration synthesis reaction, and continue adding amino acids to this chain, we would get what's called a polypeptide. So each of these dots represents a single amino acid. So for example, this first red one might be methionine, so I'm going to say it's met. The next green one might be phenylalanine, so all right, phe. The next one could be alanine, ALA. The next one could be valine. Holding each of these amino acids together, as you learned before, is a peptide bond. A peptide bond is a form of covalent bond, so it's very strong. And this whole thing together, this just long chain of amino acids all strung together through peptide bonds, is called a polypeptide. So we call this sequence of amino acids its primary structure. So the order in which the amino acids are strung together is the primary structure, the most basic level of structure of a protein. If you change one of the amino acids, then you change the primary structure of the polypeptide. Now, in order to become a protein, this polypeptide needs to be folded in a very specific way. And there are two levels of structure that we're going to call secondary structure that I want you to know for this class. But know that there are many more levels of structure beyond just these two. So the secondary structure is the first level of folding. So I'm going to draw this in have two different images for you to look at. First is just my little dots all coiled up together here, and then here is a schematic taken from your textbook or a textbook of this first level of folding. So the first level of folding that we're going to talk about is called an alpha helix. An alpha helix, as you know, I'm sure from the structure of DNA, is kind of like a spiral staircase style pattern. And the way that this structure is held in place is through hydrogen bonding interactions between the amino group of one amino acid with the carboxyl group of another amino acid. The oxygen part of the carboxyl group. So if we were to draw it here, which is maybe not going to be very effective, we would draw little hydrogen bonds holding together this structure like so. So these dotted lines, they represent hydrogen bonds. And as do these dotted lines here. So hydrogen bonds, as you remember from last semester, I'm sure, are a lot weaker than covalent bonds. So the primary structure of a protein is a lot stronger than the secondary structure. So this alpha helix is one type of folding pattern. Another very common folding pattern is called a beta sheet. So a beta sheet is when amino acids are folded back one chain on another to make kind of a flat blanket-like shape. And again, this structure is held together by hydrogen bonding interactions between one row in the sheet of amino acids to the next row of amino acids in the sheet. So this is held together by hydrogen bonds. And over here, you can see it as well. So hydrogen bonds are holding together our secondary structure. And our secondary structure is a lot more tenuous than our primary structure because the covalent bonds that hold together the primary structure are a lot stronger than the hydrogen bonds that hold together the secondary structure. Okay, the next layer of folding is called tertiary structure. And tertiary structure is going to be a combination of a couple different secondary structure motifs. So secondary structure, we would only look at like the alpha helix region or a beta pleated sheet, but a tertiary structure is going to have both in the same enzyme. So here, for example, we have an alpha helix. And over here, this other part of the enzyme, we have a beta sheet. Okay, so, and then I've kind of drawn a schematic as well. So here we have an alpha helix region of my hand-drawn enzyme right here. And here we have a beta sheet region of the enzyme here. Now, tertiary structures are also mostly held in place by hydrogen bonding. The different R groups of amino acids, we're going to look at this in class tomorrow, but they have different characteristics. And some R groups are very hydrophobic. So tertiary structures usually have R groups, hydrophobic R groups, towards the center. Because enzymes are located in aqueous solutions, meaning watery solutions, so they're going to be surrounded by water. So they often take their water-hating side chains, their hydrophobic side chains, and they point them towards the center. So that means in the center of this protein, there are probably going to be some amino acids that are very non-polar. And then our hydrophilic and polar side chains are going to face out. So this hydrophobic interior and hydrophilic exterior also helps hold the overall tertiary structure in place. So hydrogen bonding, as well as these associations of hydrophobic interior and hydrophilic exterior, help hold together the shape. And then the last thing that holds together the shape are bonds called disulfide bridges. And these are bonds between two sulfur atoms. And this is a covalent association or covalent bond. So it's going to be strong. So what's holding together tertiary structure is this hydrogen bonding, and then the kind of layers of hydrophobic interior and hydrophilic exterior, and these are all relatively weak forms of holding the structure in place, but a disulfide bridge, which is a covalent bond, which could form between two enzymes or two amino acids like this, is a strong association, and that's going to tightly hold the tertiary structure in place. But most of the structure is, again, held together by weak or weaker bonding interactions. Okay, so tertiary structure, as I said before, is two or more folding patterns. I should write that down. Okay, and the last layer of structure is called quaternary structure. And what differentiates quaternary structure from tertiary structure is that it's composed of multiple subunits, so multiple primary structures. So for example, if we were to dissect this quaternary structure protein, we would see that this is one subunit. So it's a long chain of amino acids, and we could follow it from one end all the way to the other end, and there would be no breaks in the peptide bonds holding this whole structure together. So on its own, it would be a tertiary structure protein or enzyme because it has two or more secondary structures. It's got a beta-pleated sheet and alpha helix. However, because we have multiple subunits, so here's another independent subunit right here, because there are multiple subunits, it becomes a quaternary structure protein. So a quaternary structure protein is going to have multiple subunits. Okay, and primarily what holds together quaternary structure proteins, so one subunit to the next, is hydrogen bonding interaction. So at every place where these two, or in this case 4 subunits come together, we are going to see hydrogen bonding. So this is all hydrogen bonding. Okay, all of this is to say, so primarily, if we were to look at from primary structure, which is covalent bonding, but through quaternary structure, where secondary, quaternary, and tertiary structure are all held together by hydrogen bonds, and hydrogen bonds are relatively weak, we know that if we disrupt hydrogen bonding, then protein structure will fall apart. So if we break the hydrogen bonds that hold together the quaternary structure, or the tertiary structure, or the secondary structure, we will go back to a long straight chain of amino acids, the polypeptide, and therefore the enzyme will lose its function because an enzyme has to take on a three-dimensional shape in order to perform its function. So if we disrupt hydrogen bondings, interactions, then the protein structure, the protein will fall apart and it will stop working. And we're going to see that in class tomorrow when we mess around with the proteins in the liver. Okay, that's all.